Hi, my name is Chase Baker.
And my name is Chase khedmatgozar. First off, we would like to thank Cal State East Bay for hosting this. Our project is titled Screening the Effects of Brain Targeting Compounds on Sensory Behavior and Decision Making in C. elegans. Humans are able to take in a lot of different stimuli at the same time and make a decision based on this. This is called multisensory behavior. Despite knowing that humans are capable of performing multisensory behaviors, and we're certain of certain regions in the brain, we still don't know the underlying mechanisms behind it. So if we don't understand multisensory behavior in a healthy person, we especially don't understand it in a person with a disease. There are several diseases in humans associated with problems with multisensory behavior, such as depression, bipolar disorder, and a lot of others. There's a lot of research being done in treatments for these disorders. A few examples here are lithium and valproate for bipolar disorder, and clozapine and haloperidol for other mood disorders. Despite understanding that some of these drugs are able to treat multisensory behavior disorders, we still don't know the underlying mechanisms behind these drugs and the targets of these drugs. In order to further understand the mechanisms of these drugs and how they interact with the nervous system, scientists use model systems such as mice, fruit flies, and C. elegans, which are those microscopic worms at the bottom of the screen. In our lab, we use C. elegans, and that's because they have a similarity at the level of the genome with humans. Their nervous system uses similar neurotransmitters as humans. There's mutant worms for almost every single gene, and behavior can be easily quantified. Chemicals in the brain, such as serotonin, dopamine, glutamate, and insulin, are essential for multisensory behavior and decision making. Worms have genes that encode these pathways in their nervous system too. So worms utilize the same signals in a similar manner as humans. These types of behaviors in worms are how a worm moves, lays an egg, eats, but also more complex behaviors like how a worm learns. So because of these similarities, worms are able to be used to test for potential targets of pharmacological drugs, and scientists before have used drugs to test behaviors in C. elegans. So you might be wondering, how do we test a worm's decision-making behavior? How we do this is that we first place 20 to 25 worms on the middle of a food patch. So worms won't leave the food unless they have to. We let the worms acclimate for about an hour, and after that hour, we put a dangerous chemical called nonanone, and worms hate the smell of nonanone. And now they have to make the decision. Do I stay on the food and possibly die from this dangerous chemical, or do I leave the food and possibly starve? This is the decision that we measure, and we measure their food leaving rate in 5-minute intervals for 45 minutes. This video shows worms right after the dangerous chemical nonanone was dropped. And as you can see, almost immediately the worms go to the edge of the food, and now they're making the decision. Do I stay or do I go? Do I stay or do I go? And after about 15 to 20 minutes, almost all the worms have left the food.
Here's a look at some of the drugs that we tested on our model organism, measuring the Nonanone-Dependent food leaving behavior. The way we add these drugs to our experiment and into the worms' bodies is by adding the drugs into the agar mixture that the worms are placed on during the experiment. We chose lithium specifically to focus on because of its neurological targets and being related to mood disorders such as bipolar disorder. Lithium is known to target similar pathways found in humans and in our worms. Some of these targets include dopamine receptors, glutamate, NMDA receptors, and GABA receptors. So what we found when adding lithium to our wild-type worms is that lithium enhanced the food-leaving behavior in our assay. So in the chart that you're looking at here, this is a representation of our food leaving versus time. So worms in the first 15 minutes leave quickly, and that's our target zone that we're usually focusing on. And what you can see in this chart is that lithium sped up the wild type worms and made them leave food faster than they normally would. So what we're looking at in these two charts are different concentrations of lithium on our wild type worms. The significance of these two charts shows that changing the concentration of lithium has little to no effect at the increased leaving rate that we're seeing on a wild type worm when the repellent is placed next to them. So the question we asked ourselves is, why is lithium increasing the rate of leaving the patch of food they're placed on when given lithium? So we did this experiment called a square plate assay, and you're looking at the data here for that. And so in the square plate assay, we ruled out that they don't have an increased sensitivity to the nonanone, which is the repellent. And so that leaves us with one scenario, that it has something to do with the sensitivity to food. So we know that lithium speeds up a wild type worm in this assay. In the chart in the top right that we previously looked at, you see that blue line on wild type worms on lithium, leaving faster than they normally would without lithium. So we have a handful of mutants that we've previously studied to be delayed food leavers in this assay. EGL3, DAF1, DAF7, and INX1 are the names of these mutants, and they lack the function in neuropeptide processing, TGF-beta receptors, and gap junctions. So the first mutant that we tested was EGL3. And EGL3 is the delayed effect that I was just talking about. In orange, you can see that delayed food leaving effect. When given lithium, it absolutely rescued that behavior of delayed leaving and restored it back to a normal leaving rate. So the next mutant that we tested was DAF7. Seen in orange, you can observe the delayed food leaving behavior. Once given lithium, the behavior was rescued back to a normal wild type food leaving rate. So our next mutant that we tested was DAF1. This is another delayed food leaver. Seen in orange, you can see the delayed behavior. And once given lithium in the blue line, you can see that it restored the rate back to a wild type food leaving behavior. This next mutant, INX1, is a gap junction encoding gene. This is normally a delayed food leaving mutant as well, and once given lithium, this was not rescued. This is just an example of a mutant that could not be rescued by lithium. So let's summarize all that data that we just looked at. In a normal wild type worm without lithium, they leave at a normal rate in this assay. When we look at our mutant worms without lithium, they're known to leave the food very slowly. But when these mutant worms who are delayed food leavers are given lithium, the mutant worms now leave at a normal leaving food rate. And so the significance here is that mutants lacking brain signals and have altered decision making can be rescued using lithium. So in summary, worms are able to perform a multisensory behavior assay. We saw lithium enhanced the food leaving behavior in a wild type worm. Mutants that were known delayed food leavers were rescued in the food leaving behavior by lithium. And these worms are a great model organism for studying the effects of drugs on the human nervous system. So the future goals that we have in this experiment is to continue to examine the effects of lithium on worm multisensory behavior assays. Secondly, to continue to examine the potential neuronal signaling pathways influenced by lithium during regulation of decision-making behavior through mutant analysis. Third, to identify the direct neuronal targets of lithium during the regulation of decision-making behavior through mutant analysis. And last, to perform more experiments in the presence of other drugs. For example, some of the drugs we will be looking at in the future will be methamphetamine, LSD, and haloperidol. We'd love to give a special thanks to Dr. Sean Kelly, Dr. Luis Sanchez, and Betty Ortiz, as well as the faculty student small grant that was funded through CSUCI. This helped fund some of our experiments and helped a lot during our research.
And I would like to thank LSAMP for help fund my research. And finally, we want to thank you all for listening.